All right, I'm going to talk about opportunities for AI in evolutionary and systems biology, and specifically I want to encourage collaboration in this area because I don't know if it's the being shy problem or something, but New Zealand is currently entirely missing from the country of people doing AI using molecular data. And the reason genomics has become a big data field is the cost. So here you can see that the cost of sequencing has dropped from 100 million US dollars to 1,000 currently. So having an, say, MBA Endeavor grant, you can use of $1 million, you can use it to sequence 1,000 full genomes. And that translates into the volumes of data that is being produced using various sequencing technologies, and it's expected to bypass Twitter and astronomy, which are currently the largest data producing fields soon. So methods should definitely be developed there. And although the cost of sequencing is very cheap, it's actually the data analysis that is becoming far more costly to do. And 1 million Endeavor grant is definitely not enough to analyze what you can sequence for that money. So the couple of areas I'm working on with my collaborators and students are related to phylogenetics and fitness landscapes. And here you can see the world transmission tree of SARS-CoV-2 virus, so the current pandemic. And yeah, unfortunately, I cannot show it in real time, but you can play with this Next Strain website and see how sequences have been becoming available when they arrived from different countries. So the tree was growing, and currently it has several millions of leaves. And our current algorithms are definitely not up to the job to handle these massive amounts of data. And furthermore, when you have this tree constructed with several million of leaves, and a bunch of sequences arrives from New Zealand or India or Australia, can you update the tree without rerunning the algorithm? So there is a whole big potential here for online machine learning methods that are currently not available anywhere. So epidemiology is one example of these methods being applicable. This is another example that is related to cancer evolution, and what you see here is a tree constructed on single-cell sequencing data from a single patient. So that was from an experiment. So we took cells from a patient, seeded in mice, and let it grow, and then took single-cell samples and then sequenced them to understand the evolutionary history. And when we can understand or infer the evolutionary history from sequencing data, we can tell what was the driving cause of cancer and what are the targetable mutations. And as we speak here, that's a collaboration with Southern District Health Board, and one of such analysis has actually been used in clinic, and as we speak, it's being presented to a patient with a suggestion to a few possible therapies when the currently tried ones didn't work. And yeah, another area of research in big molecular data is fitness landscape. So basically, rather than inferring trees from sequence data, you might be wondering where evolution is going to go from there. So when you have a bunch of sequence or temporal data from cancer, for instance, or from virus, you can ask, what is the next resistant strain, or what is the next resistant mutation of cancer that will lead to unpredictable outcomes. So analyzing this currently is done excluding gene-gene interactions from the analysis at all, from the models at all, and that performs really poorly. And we do think that machine learning approach such as attention-based architectures of deep neural net can help with answering these questions and bringing in interactions into the picture, which are extremely important, as we know from the biology. You know, the disease is never driven by a single gene, complex disease such as cancer. So we really need to understand what's going on. And with that, I'd like to thank my collaborators, students, and postdocs, and the lovely people who funded this work. Thank you.